Welcome to the iWorld's webinar series, Bioscan Illuminating Biodiversity. Our presenter today is iWorld's Science Committee member, Dr. Susie Wood, a senior scientist at the Cawthron Institute in Nelson, New Zealand, where she co-leads the Lakes380 project using paleo-environmental reconstruction and molecular techniques for analysis. Her webinar, Lakes380, Learning from the Past to Protect and Guide Lake Restoration, will highlight how she is using DNA barcoding and metabarcoding to assess the historic and current health of lakes in the Aotearoa-New Zealand. Susie has a PhD from Victoria University in Wellington, where she focused her studies on cyanobacterial blooms and toxins. She leads and contributes to multiple research programs, supervises students, and regularly consults on projects for government departments, regional authorities on topics related to cyanobacteria, water quality, molecular ecology, and biomonitoring. She also advocates for incorporating molecular tools in biomonitoring and biodiversity projects. Welcome, Susie, and take it away.
Tēnā koutou katoa, ko Susie Wood tōku ingoa. So good evening, good morning. I'm Susie Wood. I'm a freshwater scientist and a molecular ecologist based at the Cawthron Institute in Nelson, New Zealand. So thank you for the wonderful introduction, and thank you all for joining today. It's just a real privilege to be able to present our research to this community. So I'm going to be talking today about the research of myself and colleagues over the last 3 to 4 years, which is aimed to protect and restore the health of lakes and their biodiversity in Aotearoa, New Zealand. So I'm just going to start with a little bit of background on Aotearoa, New Zealand. So we are a small island nation in the South Pacific. We have a population of around about 4.8 million, and we are one of the youngest countries in the world, and actually one of the last places in the world that humans arrived and settled in. So settlement of humans, or the first humans, arrived only about 700 to 800 years ago. And we've been isolated from any other large landmass for around about 80 million years. And one of the unique things is that we have almost no mammals in New Zealand. So with the exception of a few small bat species, we're free of all mammals. And this has allowed a really unique flora and fauna to develop. And perhaps the most well-known of these are our charismatic large flightless birds. And pictured there, we've got our national bird, the kiwi, and next to it, the kakapo, which is the largest flightless parrot in the world. And of course, both of these are endangered now, with less than 200 kakapo alive. We've also got wonderful flora and many endemic insect species. But the focus of today's talk is on lakes and their health and biodiversity. And so globally, lakes occupy about 1% of the Earth's surface area, but they are disproportionately rich in terms of their biodiversity, with around about 10% of the world's biodiversity in our lakes. So what do we know about lakes in Aotearoa, New Zealand? So we have 3,800 lakes in Aotearoa, and the blue dots on this map show the location of these. And these are lakes that are larger than 1 hectare in size, so about the size of a soccer or a football field. And these lakes are exceptionally rich in every way imaginable. So from our largest lake, Lake Taupo, which is over 600 square kilometres in size, to our very small alpine tarns nestled up in at several thousand metres in our mountains. And these lakes, we've got, for such a small country, we've got a really huge diversity of lake types. So we've got glacial lakes, lakes that have been formed by natural damming events, volcanic lakes, dune lakes that are formed by sand blown formations. And so you'd think with such a rich and diverse set of lakes that our scientific information or our scientific knowledge on these lakes would be equally as rich and diverse. So this map again just shows the location of 3,800 lakes. What I'm going to do now is show the lakes that we currently monitor or have scientific information on. And so it's less than 2% of New Zealand's lakes that we know something about scientifically. And when I overlay the location of all lakes in New Zealand, you see that there's huge parts of the country where we have next to nothing or no information scientifically about the health of those lakes or the biodiversity of those lakes. And of those lakes that are monitored, monitoring records are pretty short. So we've only been monitoring our lakes for perhaps 1 or 2, or in the best 3 or 4 decades. And those monitoring records usually start after lake deterioration has occurred. So we don't know anything about what those lakes might have been like before human actions caused their deterioration. And also most of those lakes are in lowland, highly modified catchments. So we know very little about our alpine lakes, for example, or our lakes which have still got fully forested catchments around them. So this lack of knowledge on New Zealand's lakes and their biodiversity led my colleagues and I to come up with a project called Our Lakes Health Past, Present, Future, also known as Lakes380. And we are very fortunate to get this funded by our government about 4 years ago. So this is an extremely ambitious project, both scientifically and logistically. And what we set out to do was to sample around about 10% of New Zealand's lakes. And at each lake, we collect water samples, surface water and surface sediment samples to characterise the current biodiversity and health of the lakes. And we also collect sediment cores, and this allows us to explore how and why the health of our lakes has changed. And there's also a large social science and cultural aspect to the project, which I'll mention briefly later on. So I've just got a short video to highlight, or to give you a bit of an introduction into Lakes380. New Zealand has 3,800 lakes that are greater than 1 hectare in size. And when we look at how much scientifically we know about those lakes, or how many of those lakes we monitor, it's less than 200 of them. And those lakes that we do monitor, the monitoring records are only about 10 or 20 years long. And so we know nothing about what those lakes were like prior to human arrival in New Zealand. And so I guess the genesis for Lakes380 was this real lack of scientific information and this inability to be able to help design restoration programs.
We started the project, we were doing some pilot work on a lake over in the Wairarapa, at the bottom of the North Island. So we'd been monitoring that lake for a few years, and we'd noticed that it had been having these algal blooms in it. So we were trying to develop a pilot project to understand why that might be. But we devised it that we would look at what was happening in the modern system, so in the lake that's going on now, but we would also use the mud cores from the bottom of the lake to look back in time to see that if that had been a naturally occurring event or whether that was something more recent.
Of our lowland lakes, probably somewhere in the order of 50% of them are deteriorating in their health. So the aim of Lakes380 is to characterise the current and historic health of 10% of New Zealand's lakes. And we're doing that by collecting water and sediment samples to characterise what lives there now and what the water quality is like. And then we're also taking sediment cores. So sediment cores are like a storybook back in time, and we can go back and analyse these cores and learn more about what our lakes were once like and how and why they've changed. We're working with iwi right the way across the country, and there's 2 different components to that. One is making sure that we're sampling lakes that are culturally significant, that we're not sampling lakes that are wahitapa or sacred, and that we can work with them and share some of the information that we're gathering, but also hear their stories and histories of the lake, because they are equally as important. And when we combine those different knowledge streams, we can have a much more enriched knowledge of our lakes and their health.
So so if we can get 2 from each horizon, and then again it gets a bit messy, but you'll flick it out. I think the main hope is a better understanding of lakes, their history and what has brought them to where they are now. And also a better understanding for New Zealanders of the lakes that we have and appreciation of them, so that we can manage them in a lot better way moving forward.
My role in this project is to investigate past climate change and see how climate change impacts lake water quality. So what's fantastic about the Lakes380 project is we're in the process of collecting literally hundreds of sediment cores from around the country. And from this network of lake sediments and lake sediment cores, we can use them to get a better understanding of how climate has changed during the past millennium.
There's 8 different organisations from New Zealand involved in the Lakes380 project. They involve universities, and it's led by Cawthron Institute and GNS Science. And then there's a whole group of international collaborators that are working on the project. The team now would be something over 70 researchers.
Working with all those diverse people, great colleagues and amongst that as well, is really inspiring. Everybody has a passion for what they're doing, and to be able to do that at such a scale, on a national basis, it's a globally unique project in that way. There's nothing like it being done anywhere else in the world to the scale of what we're doing. So that as well brings it all together to make it really neat and interesting.
So hopefully that's given you a little bit of a background to the project. As I said, we're in year 4 of the project now. And so we finished the field sampling campaign, and we ended up sampling around about, or just over 310 lakes across the country. And you see some images of these wonderful lakes, and it was a real privilege to travel to them and see these lakes, and also to meet the local iwi or tribes that have long affiliations with these lakes and the communities associated with them. And so we are now really in the data analysis, sample analysis phase, and we have literally hundreds of thousands of samples. We have something like 4 kilometres of sediment core in our freezers and fridges. And as part of the social science and cultural part of the project, a lot of filming and interviewing has been done, and now there are many outputs being produced, and these are not always sort of aimed at the scientific community, but also at the general public and other community groups as well, and encourage you to look at our website to see the diversity of things we're producing. So for today's talk, I really just wanted to focus on 3 aspects of the project that I hope will be of interest to this audience. And the first one looks at how we can use environmental DNA to complement our current approaches and understand more about the spatial national scale picture, I guess, of lake health and water quality. And then I want to take you on a trip back in time and show you how we're using environmental DNA to look at the past biodiversity of our lakes and the catchments around them. And finally, finish with an example of how, as part of this project, we are working with iwi or tribes and interweaving different knowledge streams to really give us a more enriched understanding of our lakes and their importance. So starting with the issue of current water quality, and as I highlighted in my introduction, we really have quite a limited understanding of the health of lakes at a national scale in New Zealand. And so one of the great things about the Lakes380 project is that when we sat down at the start, we wanted to make sure that the lakes that we selected to sample crossed a wide range of every environmental and gradient we could possibly cover. So from land use to size and altitude, I've just picked 4 examples here. And in pink, you see that all lakes in New Zealand, and blue, you see the lakes we selected as part of our project. So really set out to collect a representative sample set, so representative of all lakes in New Zealand, which, as you'll see later, allows us to do some really neat spatial statistical modelling. And so one of the current challenges with assessing water quality is that to do this requires going to a lake, taking a water sample and analysing the nutrients and algal composition of that, but doing that every month for at least 3 to 5 years. And from that, we can calculate what we call a trophic level index. Essentially tells us whether the lake's in good, poor, very bad condition. But of course, that's a very costly and time-consuming thing to undertake, and really isn't possible, particularly in many remote locations where you might have to helicopter to them and with limited resources. We set out to ask ourselves, was there a way using environmental DNA that we could overcome, or we could develop a method that would at least complement and overcome this and allow us to assess the health of many more lakes. And to do this, we've used environmental DNA, and we've taken a sample from the surface sediment. And so this surface sediment sample is a great time integrator. So it's capturing what's happened in the lake over the past months, weeks to months to years, rather than a water sample, which tends to be more of a snapshot of what's happening in the lake at that current point in time, which can be influenced by environmental and climatic variables. And using statistical approaches, we were able to identify indicator bacteria. So bacteria that were only present in good or bad lakes, and develop up what we're calling a surface water bacterial trophic indicator. And so originally we designed this using data from 100 monitored lakes. So we collected our samples from that and selected our indicator species. And this graph just shows you that there was a very strong correlation between the trophic level indexed, which you can see here on this axis here, versus our surface bacterial trophic level index. So we're extremely pleased with the model that with the approach that we've developed here. And we then applied it to all of the samples that we had at the time, which was 255. And it started to give us a really good indication of the health of these lakes. And yeah, most worryingly, what stood out from this was that around about 48% of the lakes that we'd sampled were in poor or very poor condition. And we know that unhealthy lakes is also usually a very bad sign in terms of biodiversity and the stresses that will put on our native species. So to take this now a step further, we've just completed some spatial statistical modelling using this data and the environmental information that we had on all lakes in New Zealand to predict what lake health looks like at a national scale. And this is the result of this. And so it mirrors actually quite nicely what we saw in the data from the 255 lakes, and it really highlights the importance of having that representative data set when you're building these models. So again, this number here is about 44% of our lakes are predicted to be in a poor or very poor condition. And you can see on this map here just the location of those. So actually, if we just look at the North Island of New Zealand, which tends to be more populated and has a lot more agricultural land use, 81% of lakes in the North Island are predicted to be in eutrophic or worse condition. And we're fortunate that many of our lakes down in the South Island here are actually part of large national parks, and because their catchments are protected, fortunately they're still in very good water quality. And what we're really focusing on this data set now is these lakes that are in mesotrophic or average condition. And really there's an emphasis now to look at protection of those and how do we prevent the degradation of these lakes. And of course, we want to focus on restoration of those eutrophic lakes. And so this is a really valuable national scale picture that's allowing us to think about protection and restoration and where to focus our efforts. And it's really neat to have such an overall national picture at this point. Okay, so we're also able to apply this molecular tool that we've developed down our sediment cores and look back in history and understand how the health of our lakes has changed. So to give you an example, and this is still work in progress, this is a little lake at the bottom of the North Island, Lake Ponui. What we've got here is the sediment core. At the top here, it's essentially mud from the current day, and we're going back around about 800 years in time. And a reminder that around about 700 years, Maori, or the first people that arrived in New Zealand, settled in this region. And they did do some modifications to the catchment as they settled, changes in vegetation, but in this region, or around this lake, it was quite minimal. So prior to any human arrival in this region, this lake was in oligotrophic or good condition. There was some slight changes in water quality during the period of Maori occupation and before European arrival. But by far the biggest change to the water quality of this lake has occurred post-European arrival. And we attribute this to the introduction of non-native fish and also to changes in the land use, in particular agriculture, in parts of the catchment and fertilization. And so this lake is now in a supertrophic condition and experiences regular algal blooms. But this kind of information is extremely valuable when we are setting restoration targets and trying to develop restoration plans. So we know that this lake was once an oligotrophic condition. We can learn more about what biodiversity was there, and we can understand by layering other parts of the story on top of this, which I'm not showing, understand why the degradations occurred and actually target some specific actions to try and remediate that. So that's given you a really good idea of how we're using environmental DNA to understand the temporal and spatial health of lakes in New Zealand. What I wanted to switch now to was biodiversity and take you on a trip back in time to look at how the sediment cores are providing us information on previous biodiversity in lakes and catchments. And this is all work that's been done, or the work that I'm going to present is work that's been done using metabarcoding, so environmental DNA that we are then metabarcoding. And so the first lake the lake I want to talk about today is Lake Coringa. It's a lake on the west coast of the South Island of New Zealand, and it still has a very pristine catchment. And so the data I'm going to be showing you now is generated using metabarcoding, while we use the classic barcoding gene, the CO1 gene. We also use a number of other genes, and in some cases, depending on the species, multiple genes to capture the entire biodiversity. And so here I'm looking at the historic fish diversity in this lake. And just to orientate you, the sediment core is now lying horizontal, going from about 2015 when this core was taken back just 200 years in this case. And these different bars here are representing different native galaxid species. So quite a diversity of different species used to live in this lake. In the 1900s, brown trout was introduced to this lake. And you can see that here by the appearance of the red bar in this graph here. And it has quite a pronounced impact on our galaxid diversity. And you see that while there is still galaxid detected, that really that diversity has changed. So I guess sometimes these scientific trips back in time can be quite upsetting when we see that the types of consequences of these, I guess, actions that we've taken previously. But it's a good example of how how we can use metabarcoding and genetic information to understand changes in biodiversity and what we've lost, but also to give us aspirations of what we might want to restore or could restore and introduce back into some of these lakes. So that's an example from an in-lake community. We can look at many different organisms, things like aquatic plants, zooplankton, the insects that lived in the lake. And I also just wanted to show you some of the interesting and probably information that we least expected to get from these sediment cores is about the biodiversity around the lakes. And so what we find is that we actually pick up a large amount of bird DNA in the lake. And just to highlight some really neat examples that came out of this sediment core, and I've shaded out most of the biodiversity, but there's 3 species there which are no longer found in the catchment, but we see their DNA historically prior to that 1990 period. So one is the kakapo, the large flightless parrot. Then we've got the kokako, which is now thought to be extinct, the South Island species. So it's really interesting to see that it used to reside in this area. And of course our kiwis. So again, just a stark reminder of what we've lost from this area of New Zealand. And we can also, you remember that I said there are no mammals, native mammals in New Zealand, but we of course get a lot of mammalian DNA in our sediment core, and this is quite neat to be able to track the arrival of various different mammals, and in many ways most of these are what we would call pest species. And have impacts on our native biodiversity. So prior to the 1870s, before there was much European impact on this part of the country, the only mammal that we pick up is the kiori, which is the Polynesian rat, which arrived with the first settlers in New Zealand. In 1870, which aligns beautifully with the written history of this region, there was a cattle track that was put in to transport cattle, and while it didn't actually come close to the lake, it did pass through the catchment, and we start to pick up cattle signal in the lake. Red deer were introduced for hunting reasons and now a very common place in the catchment of this lake, and of course are sought after for hunting, but cause a number of problems for our native flora. And most recently we pick up the Australian possum in the sediment core. And while this has been in the country and other parts of the region for a long time, it only recently arrived in this part of the country. So a really interesting way of also documenting some of the arrival of the non-native species. And when we tie this all together, I think we'll be able to look at their impact on our native biodiversity as well. So the final part of the project that I want to talk to you is just to give you a couple of examples of the way that we're working with Māori, or the Indigenous people of New Zealand. So Mātauranga Māori is Indigenous knowledge, and we think there's a huge amount to be gained by learning from different streams of knowledges and looking how they complement, or sometimes they don't necessarily complement each other, but they tell us different stories, and just how we can learn from that and weave these different knowledges together. And so there's been two parts to this, I guess. One is that we did a lot of engagement with the Māori, what we would say iwi or tribes, prior to sampling, to make sure we were sampling lakes that were culturally significant and lakes that weren't sacred. And then we also have worked in depth with 4 iwi around the country. And this has involved a lot of relationship building and talking about the science that we're doing and hearing about their whakapapa, or their history and their connection with the lakes in the region. And there are many wonderful stories and bits of information that I think that we've been able to exchange through this process. And then we've also been thinking about how do we communicate this work, and how do we capture this knowledge and make sure that it's there for generations to come. And so I just want to highlight 2 ways that we've done that. And one is through what we're calling digital storytelling. And so there are wonderful stories to be heard and be told by the elders in particular from these different iwi. And we've captured those on film. And I'll just refer to our Lake Story website to look at those, and some of them about biodiversity. So our Aotearoa species, our treasured species, kakahi, which is the freshwater mussels, or tuna or eel. And there are also examples there where we've had the scientists and the iwi talking together about their lakes and interweaving their knowledge and just learning from each other's different backgrounds. And then we really have thought about how can we take all of our scientific information, and instead of showing wiggly graphs and bar diagrams like I've shown, how can we make this much more visible and appealing to iwi and also to the general public? And one way we're doing this is through virtual reality. And so using our environmental DNA, such as I've shown you today, and also some of the other more traditional approaches that we use in paleolimnology, for example, pollen analysis, we develop, scientifically, we develop a picture of what the lake looked like and what the land around the lake looked like. And there are also these oral histories from iwi that we can draw on that tell us what the landscape was like. And so using these different knowledge streams, we've created 4 virtual worlds for a Lake Mawhitu. And this is actually a 3-dimensional world that you can move through, but I'm just going to show you some still images from this. It's not quite complete yet, but it'll be launched on our website in about a month's time. And so I've taken you back 1,000 years at Lake Mawhitu here, and you can see the lush native vegetation around the lake and right up to the edges of it. And the lake is a deep, clear water lake here, lots of aquatic vegetation. It's teeming with life 1,000 years ago, and we know this from our scientific reconstructions. So if we fast forward 1,000 years to today, much of the vegetation has been cleared. The lake level is lower. Of course, our data and our observations show us that this lake now has much less biodiversity. There's an algal bloom in the lake. So yeah, this is the image of what the lake looks like today. But we've also been able to work with the local iwi, Ngāti Kuata, and various other organisations that are putting a huge effort into restoring this lake and the land around this lake. And we've captured what their vision is for the future, and we can create worlds thinking about what the aspirations are going forward. And so just to show you, this is an aspiration for 100 years' time, and you can see the return of some of that native vegetation and life returning back to the waters of Mawawhitu. So this is an interactive world, as I said, and this is just a dive underwater to show what it looks like down there. And there are all these different touch points, and you can click on those and learn, mostly through interviews with the elders from the iwi or tribes about the lake and the importance of it. And I just wanted to show you one video that we've produced that is on this, and it really shows very nicely how the different knowledge streams can be interwoven to help us understand more about the lake and the surrounds. And this particular example uses paleontology, or the study of pollen, but you could equally imagine that our DNA knowledge has been interwoven with traditional knowledge as well.
Tēnā rā koutou katoa, ko rewe nuna mahau no te tai tokirau, e mahi ana hau ki te wharewananga o Wikitoria. There's a lot of talk about so-called Western science or conventional science and mātauranga Māori. We should recognise that they are different worldviews, they have different methodologies, but those differences are telling us something as well. With science, it's all about objective observations, right? Detaching the personal from the target observations. Whereas those things are really important in mātauranga. Integral to the whole process, the connection of people with place, connection with the environment. But as I say, we can also find some overlaps. And I can give you an example from Mawawhitu. Pollen, like most science, is not perfect. It's got limitations and it's got gaps. We can recognise plants, in some cases, down to species level, but in some cases we have to lump species together because the pollen grains are very similar between two different species. Take our flaxes, for example, in New Zealand. You know, the well-known harakeke, the common flax. The pollen grain is identical, or very close in appearance, to a related species called wharariki, which is a local species at Mawawhitu, and which is actually a taonga species with Ngāti Koata. So the Western science, if you like, the paleontology, can only go so far in terms of telling us what flax is growing and when at Mawawhitu. But with the mātauranga, we know what species of flax that is because Ngāti Koata have an intricate relationship with that species. And so we can put the label wharariki on the pollen grain that we see at Mawawhitu. So there's an example of where the two knowledge streams, if you like, weave together and the one enhances the other.
So hopefully that's given you a nice overview of the Lakes380 project, and in particular how we're using DNA-based approaches to explore the current and past health and biodiversity of lakes in Aotearoa-New Zealand. And I just wanted to finish with perhaps the most important slide of the project, just to acknowledge and thank the wonderful team that's put so much effort and energy into this project over the last 3 or 4 years, and here are all of their wonderful faces. And just to say thank you very much for listening, and I'm looking forward to some questions and discussions shortly.
Kia ora, and greetings from Aotearoa, New Zealand. Thank you, Susie, for your wonderful presentation. I'm Ian Hogg from Polar Knowledge Canada, as well as from the University of Waikato in Hamilton, New Zealand, where I'm speaking to you from today. I'll be moderating this session in place of Dirk, who's unable to join us in person today. And of course, we're also joined by our speaker, Susie. If you have any questions for Susie, please send them through. We'll see them on the Zoom chat, and then I'll pass those on. But just before we start, I'd like to mention a bit more of Susie's background. As Dirk mentioned in the opening remarks, Susie received her PhD from Victoria University and began working with Catherine shortly thereafter. However, she's also had a very long association with the University of Waikato, and as well as work in Antarctica, where our tenures overlapped. Susie has long been a champion of DNA barcoding, metabarcoding, and eDNA approaches for the molecular identification of both freshwater and nearshore marine biota, and is also our New Zealand delegate to the Scientific Steering Committee for eyeball. Susie has established herself as one of New Zealand's foremost experts in cyanobacteria, as well as on water quality and harmful algal blooms in lakes and rivers. And it was on this basis of her impressive track record that led her to co-leading this very impressive $12 million 5-year project that she's presented to us today. So thank you again, Susie. And if you haven't done so, please start sending your questions through now. I'll have a look at the chat and see what we've got. The first question I have, has finding traces of terrestrial organisms, eDNA, like birds and mammals in your aquatic sediment samples made you consider adding other land-based sampling methods like malaise traps to provide a broader range of environmental data for the surveyed lakes?
Thanks, Ian. Thanks for the wonderful introduction, and yeah, great to see you again. Yeah, so that's a really good point, and it would be something that would, I think, going forward, we'd love to consider. So the focus of this study has really been on the lakes and the organisms within, but of course, it would be wonderful to consider the biodiversity. I know there are a range of other studies using those approaches happening in catchments, but we haven't paired them up with the lakes, so it's a really good idea.
Good. Actually, I have one for you, Susie. It's a comment regarding the different knowledge systems that you talked about, and it's actually quite timely for Canadian audience members. Is the Canadian government have just signed a permanent bilateral Inuit Nunangat or homeland policy with Canada's Inuit. And this represents a significant move towards a more effective partnership and collaboration covering traditional lands of the Inuit. This is very much the same concept as whenua for Maori in New Zealand. And you've already covered this very nicely in your presentation. However, if you wanted to add anything further to that, any further thoughts, or if you do see any opportunities for collaboration, say between Canada and New Zealand in this regard, yeah, I'd be interested to hear what you think.
Yeah, sure. It's yeah, I mean, it's such a crucial part of this project. And I guess some of the key lessons we've learned is that it takes time to build the sort of relationships, and it's really about those initial relationships and developing the trust, I guess, between both the research team and the iwi. And certainly, we set ourselves a mandate that we wouldn't undertake any sampling until we'd had the appropriate discussions with the iwi or the tribes that affiliated with the lakes that we worked on. And that probably took us, the best part of a year to go through that process, but it turned out to be, or it is one of the most rewarding parts of the project. And another thing that we do is when we go to a region and we're sampling lakes, we start with a knowledge sharing morning at the lake, and it's really tricky, I guess, as scientists, because often we want to dive straight in and we're under time pressure and constraints to get our samples done and get to the next lake while the weather's good. But just slowing ourselves down and taking the time at those lakes to learn from the iwi and to hear their stories and their connections. It's just been such a wonderful experience. And yeah, then in those, as I highlighted in the talk, we have 4 regions where we've really invested and spent more time developing those relationships and actually, I guess, hearing their questions and hearing what their aspirations are, and learning how we could work together to achieve some of those outcomes. And that's been an incredibly rewarding experience. And I guess it was about how we could also ensure that some of their knowledge and stories were, I guess, encapsulated in a way that would be there for decades to come. And that's how the ideas of the digital storytelling and the virtual reality, and they're also really, I think one of the challenges here is that the iwi are often now disconnected from their water bodies or lakes. So many of the tribe no longer live in the region, for example, and might be in other parts of the world. And so through these digital storytellings and virtual reality platforms, it allows them to reconnect I think that's really important in terms of getting them involved in the restoration and hearing what their aspirations are. So, yeah, but it's certainly been it's a great journey, and it's something that I'd strongly recommend taking the time to do and making it a really important part of research projects.
Great, thanks, Susie. Just looking at our chats here. This looks like a good one. What are your plans for the future of the project?
Yeah, so it's a great question. We've got about a year left on the project. And so really at the moment it's about crunching through a lot of data. And then I think what our vision would be that this information is really used to improve the management of lakes and the biodiversity within lakes in New Zealand. And so we're putting quite a lot of effort into thinking about how we communicate that knowledge back both to the general public, but also to the management agencies. And yeah, we'd love to just keep on sampling more lakes. And I think that the world of metabarcoding and barcoding is just expanding so rapidly that our possibilities now of what organisms we can detect and in these lakes is amazing. And so that's really neat to keep on characterizing our biodiversity. And I like the likes of yourself, Ian, have done a wonderful job of developing our database of sequences, but we absolutely need to keep doing that for the organisms and lakes. Yeah, I think there's huge potential, and I think that'll be a game changer as we go forward, the use of barcoding and metabarcoding to understand and monitor our freshwater environments.
Yeah. The next question I've got here it's sort of related to that probably a little in terms of getting your knowledge out to a wider audience, but what inspired the team to create a virtual world as part of the project?
Yeah, so I guess it was this, really thinking about science communication and how do we take this really dense scientific knowledge and display it in a way that would be much more understandable for the general public, for iwi. And I think it's from some of the knowledge sharing mornings that we've held, it's such a visual thing when people see a sediment core and they can see these really stark changes in the colour of it. And we talk about, well, actually that's when all of the trees were removed in the region, and actually that caused a huge sediment pulse to go into your lake, and it killed all of the vegetation inside in the lake. And so we wanted to just make some of those things more visual so people could actually understand the information that was coming out of the science. And it's a really neat, I think once it's live you'll see it's a real sort of three-dimensional world that you can move around in different views. So it's really impactful, and we hope that it's also a neat education tool that can be used by the local iwi and the local schools around here to understand how the actions of humans and what's happening in the land is impacting our freshwater systems. And yeah, I guess just the opportunity to work with Ngāti Koata, who are the local iwi, and draw on their knowledge and again allow reconnection with that lake, which is actually on an offshore island, so it's a very hard lake to get to as well. So this was a neat way to do all of those things at once, I guess.
Neat. Are there any other interesting examples you can think of where the Indigenous knowledge created by working with the Māori filled in gaps or even contradicted the established science?
Oh yeah, it's a good question. So there are quite a lot of examples. One that's really interesting is there's a native, it's a native plant, I guess, in New Zealand. We call it raupo, but it's bulrush, so people might be familiar with it around the world. There's various different species of it, and it grows in the I guess around the edges of lakes, and so the Māori used that as a for all sorts of things. They used to build boats out of it. They used it for insulation for their huts. They even used the pollen to make flour for various different like bread like sort of food that they made. And so what we've seen from our science record is that actually quite a lot of these lakes didn't have raupo in them, but around the time of Maori settlement in the region, it suddenly starts to appear in our environmental records. And so we've been able to pair that up with the oral histories, and there's these wonderful examples we're seeing where their stories actually quite nicely align, and there's some wonderful, I guess what we'd call myths that talk about the arrival of raupo in those lakes. So yeah, that's another example of, and that one's coming up sort of repeatedly around the country. Where these knowledge streams are complementing each other.
Yeah. Okay. The next one is, in the person has admitted that they may have missed it during the presentation, but how did you date the sediment cores from the lakes?
Yes, I actually didn't cover that off in the talk, but we each sediment core, when it arrives in the lab, and it gets split in half, and we have a bunch of different things that we do to it, and one of them is that we subsample it at various different depths, and we use two different techniques to look at, or to help us understand the age of each of those layers. And so one is what we call radiocarbon dating, and so we're looking for leaves or fragments of organisms in the lake that we can pull out and date based on the carbon in those. And up for the last sort of 200 years, we use a technique where we look at lead-210, and because of the testing of atomic bombs in the atmosphere, we can correlate the concentration of lead-210 in the sediment core, and allows us to determine the age. And actually the other thing we have in New Zealand is, because we understand the timing of landscape change in New Zealand, in particular the arrival of Maori and the arrival of European and the introduction of various different species, we can also use the pollen to give us an approximate age. So we sort of combine these three different techniques and develop what we'd call an age model to estimate the age of them.
All right. I'm not sure that I'm seeing any other ones come through. Oh, yeah, no, here's one. What do you do with the sediment cores after you've finished with them?
Yeah, so at every lake we actually take 4 sediment cores, and I guess that the eDNA that I've talked about here is just one part of the process, and they also get analysed for pollen. We have several scanning techniques to look at algal composition and elements that are in the core. And so some of those analyses are, I guess, what we'd call destructive. So once we've extracted the DNA, essentially the material has been used up. But certainly a lot of it has been archived, and so we have, I think, 3 now fridge and freezer 40-foot containers that store them. And so, like I said, we've got somewhere in the order of 2 or 3 kilometres of sediment core, and really what that is becoming now is a national archive that, as this project finishes and going forward, as a resource for I guess researchers worldwide to access, and as they become interested in the lake and as our techniques improve, that'll be there to call on. And it is such a sort of time-consuming and expensive process to get these samples that we wanted to make sure it was there for many decades to come. Yeah.
With that in mind, I might sneak in another one of my questions. So you in a lot of your slides, you had examples of charismatic megafauna. So you had the kiwi and kakapo, and you had the mammals. What sorts of patterns do you see, if any, for some of the invertebrate life, say, for example, going back through time, and do you see changes, if you do, in fact, look at that aspect at all?
Yeah, so in terms of the invertebrates with the DNA, it's taken us a little while to get that working on the historic sediment cores, and so we're really just starting to generate information now on things like the chironomids and the various other sort of zooplankton organisms. But certainly some of the other more traditional approaches which look at the morphology of some of the fossils that remain in the sediment core from those organisms show us huge changes. So often it is a loss in biodiversity as that lake, the water quality of that lake deteriorates, and as they become what we'd call more eutrophic or nutrient rich, there are certain species that start to dominate that are able to survive in those sort of, I guess, very extreme, often extreme environments, which are low in oxygen and high in nutrients. Yeah, and then obviously we also look a lot at the bacteria and the algal composition, and almost all of our lowland lakes, those have changed significantly in the last, mostly in the last 50 years, actually. So it's quite sad in a way that a lot of this deterioration and loss of biodiversity has really happened in our lifetime, or not much longer than that. And yeah, and so we're really starting to see remarkable patterns of that as we start to bring these national data sets together.
Great. Yeah, the bacterial trophic index certainly looked like a tremendous fit. So that was, you must have been very, very pleased when you saw that.
Yeah, no, it's great. And I guess, I mean, the dream is that we can start to monitor more lakes, I guess. And, at the moment we're so limited because of the techniques we use as to how many lakes we understand something about their health and quality. And as we start to apply that index and the use of environmental DNA, of course, hopefully we start to really get some good ideas of our biodiversity and lake health nationally. So yeah, so it's a neat time to be a scientist and part of that area of research.
Yeah. All right. Okay. Well, I think we're just coming up on the hour there, Susie. I'd like to thank you very much again for a wonderful presentation. It was really nice to see you again, and look forward to catching up. And thank you very much for all of the people that have joined us today and for the questions that have been asked. It's been a really wonderful session. So thanks again, everyone, and enjoy the rest of your days.
Thank you, everyone.